fertilization. 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 Fertilization or syngamy. We've already discussed the site of fertilization. The site of fertilization is ampulla and isthmus junction. Which is also known as ampullary isthmic junction. Ampullary isthmic junction. Ampulla and isthmus are the parts of fallopian tube. Ampulla and isthmus are the parts of the fallopian tube. The fallopian tube was something like this. Inwardly directed processes, the funnel-shaped structure. The fimbriae we studied, and then the tubular part, which opens into the uterus. The part which opens into the uterus, which connects the uterus to the fallopian tube, this region is known as the isthmus. This is the isthmus. This region is the infundibulum. This region is the infundibulum, and in the center, in between these two, this part. This part is the ampulla. This is the ampullary region. So ampullary isthmic junction is the site of fertilization. Here the fertilization occurs, right? The sperms which are released at the cervix they would travel. The sperms, they would travel first through the cervix into the body of the uterus, then up inwards and towards the isthmus. Then they would travel across the isthmus and would reach this junction, right, where the egg is present or the ovum is present. Here is this egg, the female gamete. The egg is present. The sperms. Now steps. The steps of fertilization. The steps. The steps of fertilization. The first is approximation. पहले steps के नाम लिख लेते हैं, फिर इनको आगे डिस्कस कर लेंगे. Approximation actually means that the gametes come close to each other. The sperms come close to the eggs. The egg. Second, agglutination. Agglutination means binding. One of the sperm, it becomes fixed to the surface of the ovum. The third, depolarization. the membrane of the egg becomes depolarized. The polarity is the polarity changes. Right. Then action of enzymes. Action of enzymes of acrosome. Of the acrosome. As a result, the coverings of the ovum are dissolved. The enzymes act upon the coverings of the egg, and a tiny opening is created through which the sperm begins to enter. Right. Entry of spermatozoa. The spermatozoa enters into the egg. Cortical reaction. Cortical reaction. The action of enzymes. The action of enzymes of acrosome. This can be written as acrosomal reaction. इसको हम क्या लिख सकते हैं? Acrosomal reaction. Acrosomal reaction. Right. Depolarization, acrosomal reaction, entry of the spermatozoa, cortical reaction. Formation of fertilization cone. Formation of fertilization cone. This happens at the entry of the spermatozoa. Amphimixis. Amphimixis. Mixing of the genetic material of two sides. Then cleavage. The division of the zygote immediately. Right. Sometimes this cleavage is written as a part of a step in fertilization, and sometimes this is excluded. Right. It's better to exclude this step because this is related to embryogenesis. Right. So it should be excluded. It should be excluded. The best is to study fertilization in these 8 steps. Right. First, note it down. The first step, approximation. It simply means coming together of the gametes. The gametes come close to each other. Approximation. This is preceded by, इससे पहले एक step होता है, preceded by capacitation. Capacitation. As such, ये बन जाता है first step. अभी हमने आपको कहा था, we would be studying when I would be explaining these 8 steps. So there would be in total 10 steps. So this becomes the first. This is a primary condition. This is necessary. Now what is capacitation? capacitation is inducing capability of movement syngamy, that is fusion, and fertilization, the capacity of movement syngamy and fertilization, when this capacity is induced in the spermatozoa, the spermatozoa are capacitated. Inducing capability of movement, syngamy, and fertilization in the spermatozoa is known as capacitation. It begins in epididymis. It begins in epididymis. If you remember, when we studied male reproductive system, the tract, epididymis was a 6-meter long tube which stored the spermatozoa. It begins in epididymis. Epididymis helps to release the tail of the sperm from its cell membrane, its membrane. We studied structure, spermatid. Now this is spermatid. Spermatid has got a nucleus. Now several changes occurs in this and it becomes a spermatozoa. It becomes a spermatozoa. Now human sperm has got different parts. स्पर्मेटोजोआ का डायग्राम बनाना बोर्ड्स में भी आता है, इसके पार्ट्स नीट में भी पूछे जाते हैं. The first region is the head. It is followed by a long middle piece. This is the middle piece. And through this middle piece, several filaments. These travel to the outside. Several fibers coming to the outside as the tail of the spermatozoa. Right. In this head, there is a sac-like structure present in the front. This structure is known as acrosome. This is called acrosome. Behind the acrosome is a long nucleus. The nucleus loses its spherical shape and becomes cylindrical. This is the nucleus, right? Acrosome and nucleus are present in the head region. Present in the head region. There is a small centriole extending from the head into the neck. This region is the neck. This is This centriole extending from the head into the neck is known as the proximal centriole. This is known as the proximal centriole, right? Now this region is the middle piece. This region complete is the this is the middle piece. The end, there is the tail. There is a tail, right? Now this middle piece has several mitochondria. And these mitochondria actually wrap these cover these mitochondria wrap this fibrous part. This fibrous region is actually arising from a centriole, which is present here. The centriole is giving rise to this complete tail. This centriole is the distal centriole. This centriole is the distal, पीछे वाला. Distal centriole. This is the distal centriole, right? The sheath of mitochondria. These are mitochondria, right? There is this word, Nibenkern. Nibenkern, K-E-R-N. Nibenkern is a sheath. It is a sheath covering of mitochondria. Over the fibrous part, and this fibrous cord, जो बीच में से cord निकल रही है, this fibrous cord is known as, this sheath is present over the exoneme. It is present over the exoneme, right? There's one more thing to remember. The tail is also covered by the membrane. It's a previous year AIMS question. Which part of the sperm is devoid of cell membrane? The answer is none of the above. The complete spermatozoa has got cell membrane. Just the tip of the tail is uncovered, right? If I talk about head, it has membrane. If I talk about the middle piece, it has got a membrane. If I talk about the tail, even this has a membrane. The neck is also covered by the cell membrane, right? The tip, however, is not covered by the cell membrane, right? But in that particular question, which was asked in AIMS, the question was, which part of the sperm is devoid of, I'll write the question on the next slide. Which part of the sperm is devoid of, is lacking the cell membrane? The options were head, middle piece, tail, and none of the above. In this question, the answer was none of the above, because the complete sperm is covered by the cell membrane. It's covered. There's one more thing. In this region of head, there's a very small amount of cytoplasm. Very small amount of cytoplasm which surrounds the nucleus. It surrounds the nucleus. The small amount of cytoplasm, this small amount of cytoplasm is known as manchet. This is known as MANCHE double TE. This is manchet. This is cytoplasm. The residual cytoplasm. Manchet is the cytoplasm. Right. Now this tail is actually wrapped upon this middle piece. It is, you can say, glued to this region. What epididymis does is it releases the tail of the sperm. Hence we say capacitation begins in the epididymis. इसलिए हम कहते हैं epididymis में capacitation शुरू होता है. Right? Okay. Now. Questions? Capacitation begins in epididymis. Epididymis. Second question. Capacitation occurs in. Capacitation occurs in. Now, when we say occurs in, we actually mean completion. Occurs in female reproductive tract. Female reproductive tract. The sperms are released as packets or curd-like viscous mass of semen. Fibrinolysin dissolves this mass and this mass and the mass becomes dilute. This mass becomes dilute. This dilute mixture mixes with, it mixes with mucus. The secretions of female reproductive tract, and hence the sperms become totally free and separated. सारे के सारे स्पर्म्स एक दूसरे से काफी दूर हो जाते हैं. The sperms can now swim freely towards the egg. Right? This is at the rate is 1 millimeter per minute. Sorry, it's 1 centimeter. Sorry. The rate is 1 centimeter. 1 centimeter per minute. In 1 minute, it travels 1 centimeter. The sperms can now swim freely towards the egg at the rate 1 centimeter per minute. The motion is wriggling motion. Sperm के movement को हम क्या बोलते हैं? Wriggling. Motion is wriggling motion. Complete spermatozoa is covered by, I'm just writing the answer straight away, is covered by membrane. Except tip of the tail. Centriole in the head region. Proximal. Centriole in the neck region.